Hello? Good? You can hear me? So first of all, many thanks to the chairperson. Thank you for the introduction. Thank you all the faculty, all of you lovely students and staff who put this beautiful day together. It's so far it's been such a pleasure, and just seeing all of you here. Thank you. I had a lovely lunch, and we were just joking after giving us so much food. Instead of asking me to do lecture, you should have given me a bed so I can sleep nicely. But in absence of a bed, I will talk a little bit about the research. So before we get to the microbiome genomics projects, I'll do a little bit more of a continuation of the morning session. So we'll do a little bit more of the human population histories and ancient Egypt. Because that knowledge of this is very important to understand further some of the disease aspects and anything else that many of you may be interested in. Before I start, for all the students here, can you put up your hand if you are in the science section? Any science? If you're doing science, bachelor's, okay. If you're in the arts division, can you put up your hand? Arts. Social science also. Okay, so it's almost 50-50 split. That's good. It's good for me to know, so I can hopefully cater to all your interests. And don't be shy, please. Anybody, especially students, if you have a question or if I'm going too fast, please raise your hand or just shout. Tell me to slow down, tell me to be clear, okay? Please. Because if you miss something in the beginning, then you won't understand anything towards the end. So it's better to be clear, okay? So, as in the morning session I mentioned, I am a faculty member at the University of Chicago. I'm primarily a human geneticist. So, much like many human geneticists, one of the big interests of me and my research group is to understand what makes us so diverse. You know, Dr. Rai and Dr. Raghavan, both of them have mentioned to you, giving you many examples how diverse the global population is of humans. Yet, if you think about how much DNA differences are there, as Dr. Niraj already said, almost 99% is similar between humans and other hominids. But if you look between humans, so if you take one individual from let's say Africa, and you take another individual from America or China, let's say, and if you compare the DNA, it's 99.9%. So only 0.1% DNA may differ between different people across the world. And still, you see so much diversity, right, among us. And that diversity can be many forms, right? It can be the way we look, the way we act or behave, the way we are susceptible to different diseases. So that 0.1% of the DNA and the environment and lifestyle that we lead is really what makes us, each population, unique. And so while many human geneticists think about what is driving this unique variation, people like the three of us, we think a little bit more about what has driven it. So we take a more of a past perspective. And the reason for that is that our DNA, the way we have evolved, we haven't evolved in the last 10 years or 20 years. We have evolved as a product of the last hundreds of thousands of years. So if we want to truly understand what makes us all different or similar, we have to think about what all those different processes have been in the last hundreds of thousands of years that have impacted our development, right? So this is, the science students can take a little break. So for our arts friends, just to bring you up to speed. So as a human geneticist, the sort of, let's say the data that we deal with is DNA data, right? So you have DNA in your body, you have different kinds of DNA, and I'll come to that. But mainly it's these strands of information that our body has, okay? And so we have four bases. So these are coded as like different alphabets. So you have adenine, A, thymine, guanine, and cytosine. And we, in our bodies, we have almost 3 + 3 billion combinations of these bases. And each one of us will have a different combination. Unless you are identical twin, then you have the same DNA. But otherwise, all of us will have some difference, little bit of difference. But in the general scheme of things, we're all made of the same stuff. Okay, we're all the same. And so, as I said, there are different kinds of DNA in your body, and Dr. Guy already mentioned a little bit of this. So, first of all, all of us have cells in our body, right? And inside a cell, you have different organelles. One of the main organelles is the nucleus, and the biggest part of your DNA resides inside the nucleus, right? So, you have the nuclear DNA, and the nuclear DNA is what makes up most of your DNA. So you have almost as humans, 3 billion bases of DNA. But the catch is that you'll get part of that from your mother and part from your father. Okay? So you have 2 copies of nuclear DNA, one from mother and one from father. In addition to nuclear DNA, you have another organelle called the mitochondria. The mitochondria is a powerhouse of your cell, of your body. And there is a smaller, what we call DNA genome in there. Unlike the nucleus, which I said it's 3 billion bases, so 3 billion combination of those alphabets, the mitochondrial DNA is much smaller. It's about 16,500, that's all. It's a much smaller version. But it's there. As geneticists, we read and analyze both the mitochondrial DNA and the nuclear DNA. Okay? And both will give us different perspectives about ourselves. So what are those perspectives? So the nuclear DNA, as I said, comes from both your parents, right? And in turn, your mother will inherit her nuclear DNA from her parents, so your grandparents, and your father as well. So your DNA, your nuclear DNA, is a mix of many, many generations of ancestors that you have on both maternal and paternal side. So if we are analyzing your nuclear DNA, we can get really an insight into your full generations on both your parents' side, kind of, right? But the mitochondrial DNA is very interesting because only the mother or the woman can pass the mitochondria, right? So I think Dr. Laya also mentioned this. So for example, if I have a brother, I don't have a brother, but if I had a brother, both of us will get mitochondrial DNA from our mother. But in the next generation, only I can pass my mother's mitochondria to my children. My brother would not be able to pass. So you're only able to read the maternal, your mother's side through the mitochondria. And the other version of that is the Y chromosome, as Dr. I also said. Y chromosome only women don't have. So only the male lineage will pass it. And so you get this unique and almost complementary insight depending on which genome or which DNA you're reading. If you read the nuclear DNA, you'll get both sides of your family. Mitochondria will only tell you about the mother's side, and the Y chromosome, if you're a man, will tell you about your father's side. And as human geneticists, we use all this information to make a more complete, comprehensive picture about our species history. Any questions so far? It's clear? Okay. So, this is sort of the basic organization. It's very simplified, but it's basic organization. We talk about a cell. In that, you have this sort of chromosome, which are structures. And within that chromosome, you have the DNA and ultimately the gene, which Dr. Neil also implied. And it's really the DNA strands that we're reading. As I said, each individual will have some combination of ACTGs, right? And you will have 2 copies. So here I've given 2 examples, 2 individuals DNA. This is fake. I just made it up. But you'll have individual 1 has 2 copies, 1 from dad, 1 from mom. Individual 2 has 2 copies, 1 from dad, 1 from mom. And I just wanted to give those of you who are not in the genetics field some idea of some of these, like how do we analyze this data, right? So as an example, you can see that all of this, the letters in black, those are identical across the 2 individuals. And like I said, that's going to be the case because 99.9% of all our DNA is very similar. So most of our DNA will be the same code. But it's only very few locations along the DNA that will differ. So as an example, in red, you can see this individual 1 has G and C. So they have sort of 2 different versions of that position. And because of 2 different positions, they are a heterozygote. They have 2 versions, okay? Whereas this individual has the same C and C, so they're homozygous for that location. So these are generally words that you will read in papers, so I just want to make sure that everybody knows what these definitions mean. Okay? And generally, this is the kind of data that we get in the lab, and this is how we analyze and try to compare who shares more differences and similarities between each other. Depending on that, we can understand who shares more recent common ancestors. This is a very, very simplified version, but just to give you all a picture. So when I say that I'm trying to understand the history of our species. And some of these questions came up during Dr. Rai's talk as well. So what do we know, even before us geneticists even came to the picture, there's a whole field that has been studying the history of our species. And these are the hard-working anthropologists and the archaeologists, okay? So it's not to say that geneticists are the ones who hold the key to the origin of our species. This has been, a lot of the hypotheses are coming from paleoanthropology and archaeological field. And so as Dr. Rai explained, our human lineage is very ancestral, not Homo sapiens, but generally the hominin lineages. So sometime around between 5 to 7, this is millions of years, and these are different forms of different hominins over the time. So about 5 to 7 million years, we have a divergence of the chimp and the human lineage separates, which means we have something, particularly in Africa, in fossil evidence, that sort of resembles the human family tree. About 4 million, so you have the Homo erectus, Homo habilis, and all these sort of different hominins. And ultimately, if you come to the tip, like really the present times, you start seeing the emergence of the anatomically non-humans. That Sir's question was, what is the history of the emergence of humans? So we, as anatomically non-humans, we are one of the most boring species in that we are very, very new, okay? If you think about the kind of diversity, I started by saying, oh, we are so diverse, but actually you compare us to any other species, we are actually quite boring. And the reason for that is our origins are very, very recent. So the current consensus on when modern anatomically modern humans have originated based on fossil evidence and genetic evidence is sometime around 200,000 years. There's huge debate, it could be a little bit older, it could be a little bit younger, but on average about 150 to 200,000 years emergence of our species in Africa is the current model. In addition, as Dr. Rai mentioned, we have not been alone. Particularly, everybody has heard, I think, of the Neanderthals that have gone extinct. So those were very much there, particularly outside of Africa. So when our ancestors, some of them left Africa and came, they would have definitely interacted, and I'll talk a little bit more about what that interaction was. But this is just to give you now a broader context of what is already known even without genetics coming into picture based on fossil evidence. Okay. So, when I started, I told you that people like us, we think about the past. Okay? We look at the current, the present, but we're thinking about how all of this diversity is found. And one of the examples to kind of make this more concrete as to why we should look to the past is if you think about how we've evolved our species, ancestrally, all our ancestors were hunter-gatherers. Anywhere in the world. We didn't start with agriculture, right? Agriculture comes much, much later, as Dr. Rai also pointed out. There's different centers of domestication, such as the Middle East, East Asia. Sometime between, depending on where in the world you are, sometime between about 5,000 to 12,000 years before, archaeologically, it is known that humans invented domestication and agriculture. Before that, we were all hunter-gatherers. In fact, even today, if you go to parts of Africa, parts of South Asia, like the Andaman Islands or Southeast Asia, some tribes exist who are purely hunter-gatherers. They retain the traditional way of living. But most of us, we've just eaten rice for lunch, so we're all agro-pastoralists for the most part. Even if we are not working in the fields, but our subsistence has completely changed compared to our ancestors, right? So all of this has happened in the last 12,000 years of our history. I told you that our species originated about 200,000 years ago, but in the last 10,000 years, we've had some of the most intense changes in our species lifestyle. We have entirely changed our food strategy from hunting and gathering to a very carbohydrate-rich, people started settling down into civilizations. The mobility that hunter-gatherers had, that was lost. So we've completely changed the way that our species, our ancestors life that we sort of switched to. And so when you think about agriculture, domestication, pastoralism is using animals for dairy products, right? Whether you use cattle or sheep, whatever your source is. Then in terms of migrations, we think about long distance or horse carts and all of these inventions. Urbanization, I talked about people settling into city-state civilizations like the Harappan civilization, Babylonian civilization, Egyptian. All of these have happened in the last few thousand years. So to get an insight into how this past changes have created and shaped us, we can study the current diversity all we want, but we won't be able to get that direct insight as to what happened in the past that shaped the way we are. Okay, so this is just to give you a concrete example of why we want to live in the past. In particular, we know there's many evolutionary processes. So if some of you may have studied a little bit of population genetics, perhaps. So you recognize what creates genetic diversity, genetic drift, mutations, selection, natural selection, and your gene flow and migration. So all of this has, the rates have changed over time. In fact, over time and space as well. So there is a need not just to look globally, but also in the past to get this unique temporal perspective, right? So this is why we go to ancient DNA. Again, hopefully after these 3 talks, you are now convinced why we need to, why ancient DNA is exciting, why it's important, in fact. As this is already in your poster, this is a very famous cartoon, and I really like it because this is truly, I think your principal also this morning mentioned the importance of interdisciplinary research. And I think ancient DNA is one very unique field that requires so many different expertise to come together. And I think this cartoon really shows that, there's these archaeologists who are digging up, obviously not DNA, but some bones, but those bones will have DNA and proteins and other things that we can examine in the lab. So, Dr. Nagarjuna went through a little bit of the technical details, but I thought I'll give you a little bit more as well. So, for example, when maybe Professor Milton or some archaeologists will bring some ancient bones to us, or teeth or whatever, it's not going to be as easy. Some of you may have done DNA extractions. Have any of you done? Raise hand, or how to do, So in the class, you may be taught, okay, you can take blood, saliva, hair from all of us, or anything you can take organic material, you can extract DNA. If you extract DNA from us, like if I go around and ask you all to donate your hair or saliva, and I go to the lab and I take out DNA, almost more than 99% of that DNA will be yours, okay? Because I'm getting fresh material from you. But if I get a piece of bone from Professor Milton and I take it to the lab and extract DNA and read it, majority of that will be that poor old ancient human who died at that time. And the reason for that is that poor individual skeleton has been lying in the sediment for tens of thousands or hundreds of years. And so it's attracting all this, what we call contamination. So mostly it will be all these bacterial, soil bacteria, will just get absorbed by the bones. Over time, the bacteria will chew up all the DNA, it will degrade it. So it's really not an easy job for us doing ancient DNA. It's extremely difficult and extremely can be expensive also. Because for the same amount of money, from fresh samples we can get a lot of DNA, but from ancient samples we may get very little DNA. So we have to do a lot more DNA sequencing to get enough data to actually do meaningful analysis. So it's a very tough and risky field, but it's very rewarding. If you get the DNA, then nothing like it, right? So if any of you become interested eventually in read papers, you will encounter these words endogenous DNA. So endogenous means anything that's native, so that bones the human DNA from that ancient human being. Whereas exogenous or contamination would be all the other things like soil bacteria, or it could also be when archaeologists are digging up, or maybe in the museum where samples are being stored, and if you go and touch with your bare hands, then the archaeologist's DNA or the museum person's DNA, that will all be deposited on that. So that's all we consider contamination, because we are only interested in the ancient person's DNA, not in my DNA or Professor Milton's or Dr. Nagarjuna's. We know our DNA. We know that old DNA. So all of this makes ancient DNA very, very challenging. But we can still work around this. So when I said DNA damage, I won't go too much into details. I know some of you may be biochemistry interested, so I just put this for those of you. But mainly there are two kinds of damage to DNA, and I think Dr. Niraj mentioned this. And these two are important for us because A, they make it difficult for us to do the analysis, but on the other hand, if we see these two kinds of damage in our data, that means we are sure it's coming from an ancient source and not modern source. Because modern DNA will be fresh, it will be intact, there will be no degradation. Okay? But ancient DNA, because of the soil microbes, pH, humidity, all of that, will start degrading. And so what are those two kinds of degradation patterns? One is nicks or repeat. So nicks basically means fragmentation. So the DNA will be in our system very, very fragmented, kilobases, right? Hundreds and thousands of DNA bases are intact. But with ancient DNA, it will be very, very short, like 100 bases, like 100 letters, that's it, 100 to 100, that's it, because of all these degradation pathways. And the second kind of damage is something called misfolding lesions, and this basically means there can be certain parts of the DNA that will just mutate again because of these environmental processes. In that case, it becomes very difficult because when we are analyzing DNA data, we can look at that mutation and we can think it's a true biological mutation. But actually, it may be because degradation process. So that's why we have to be very careful when we're analyzing our DNA data, so we don't misinterpret something based on the contamination. It has to be clear. So, like I said, there are some things we can do. Some things are out of our hands, like we cannot control soil bacteria. They are there. They've been there for many millions of years. They will continue to degrade the DNA. But for new excavations, for example, and we have worked, I know Dr. Niraj's team has worked, my team has also worked, we train the archaeological teams to work kind of like this. So you can see, I think all of you now after COVID are very familiar with PPE gear. So in ancient DNA labs, we always work full PPE gear. We cannot work like this. We have to put full body suit, gloves, face mask, because otherwise we are going to contaminate further our samples, right? So in fact, if the excavations are fresh, it's nothing like it to train our archaeological collaborators or historian collaborators to be as careful, at least put gloves and face masks so their DNA is not shared on the samples that they excavate. Okay? So we can do that, and that's called highly controlled sampling. Now unfortunately for us, we cannot do this for samples that have already been excavated because that's too late for that. So for those, we can, for example, consult with those who have the samples. They may be storing it in not good conditions. So for example, your post-excavation storage conditions will also make a difference. If you just leave it like this in 50-degree weather, it's going to degrade very fast. So you have to have, again, a cool environment, less humidity environment, and then that way you can preserve it even after excavation processes. The other thing we can do, and we should do, most of us work in engineering lab, like I said, even after all of these precautions, we could accidentally contaminate a sample with our own DNA. So for that, it's good that you know what your DNA sequence is, and that way you can compare with your data, make sure it's not the same. It's not my DNA data that's there. So there are a few things we can take in terms of precautions. The last thing we do, as I said, is this is a lab photo of my lab. It's very similar to Dr. Niraj's lab in Lucknow. So like I said, we cannot work in any lab. Like any modern DNA facility, we cannot work. We have to build a brand new facility which is extremely sterile, like a forensics lab. We have to wear PPE, we have UV lamps, like you have these lights, we have UV lights. Every night for about 6-7 hours, it will come on, it will decontaminate everything. We have to bleach all the surfaces to make sure that, again, contamination is not happening. And we have to, here you can see there's a, maybe not as steady, but here's a wall. There's 2 rooms and there's 2 people working. I don't know if you can see, but there's 2 people working. And so it's also like in modern DNA labs, I can sit next to my friend and we can happily chat and we can do our DNA extraction. Ancient DNA, we cannot do that because we may cross-contaminate each other's samples. Already we have a lot of degraded material. On top of that, why risk? So we have to work separately. So it's a very, very, different mindset when you work with ancient DNA. So a lot of us came, my training was in modern DNA, so I said, oh, I know how to do this. But then when I started my PhD, they had to retrain me because then I had to think about this forensics type, sterile kind of environment. So it's very interesting, also very exciting if you get to work with this. So, yeah. So the reason initially I asked all of you students why, where, which fields you're all from is I want to bring it back to this that in ancient DNA, we think about it as a field, but actually it's a coming together of basically all three disciplines, right? We are all biological scientists. We come from genetics or molecular biology or biochemistry. We are using all those procedures to get the DNA to analyze the DNA. But where are our samples coming from? Where are the contexts coming from? They're coming from our social science collaborator, whether archaeologists, anthropologists, linguists. Furthermore, we also have to keep in mind the soil chronology. How is the soil being formed? So that will inform how well it will preserve samples over time, how the soil pH, soil humidity. So geochemistry is also very, very important in ancient DNA. So it's a really, really highly collaborative field, and if some of you enjoy that collaborative nature, then I invite you to explore graduates who are in this field, because I think you'll really enjoy it. So I skipped this because Dr. Nagarjuna did a really talk. This is just showing various sources of ancient DNA. Mostly if you're studying in human ancient DNA, most of the samples will be bones or teeth. Sometimes we work with hair, but hair is not that rich a source because the hair will undergo apoptosis and there's very little cellular matter. Bones and teeth will be the ideal, and that's also what preserves most archaeological contexts, right? So very quickly, just to make this more interactive and make sure the students are not falling asleep, this is a quiz time. So this already you have seen, and I've already given you actually results. So can someone tell me, so I'm going to show you in the next 3 slides 3 different parameters, environmental parameters. One here is temperature, and then we'll come to the next 2. So in terms of temperature, can someone just tell me what is a good preservation environment for DNA long-term survival based on this graph? Cold. Your friend has said cold. Who agrees with her? Who says cold is good? Raise your hand. So most of you think hot is good, or most don't have opinion. Most of you are falling asleep, no? Who says hot is good? You say hot is good, okay. So how do you interpret this? So this is DNA survival, okay? So the higher you are, that means more DNA survival, okay? So here on this side of the graph, it's cold temperatures, and this is hot temperature, okay? So you can see in the cold temperature is where you have maximum survival. So it will be colder. Colder the better. Okay? So same way, here is the second parameter, humidity. What do you think?
Less humidity.
This is less, this is dry, this is humid. Less humidity. Everybody agrees? Good. So if we put these two results together, cold and less humid, what do you think are the prospects of ancient DNA in a region like South Asia? What is South Asia? Tropic, hot and humid. So, again, in addition to all the challenges I told you already, contamination, degradation, in a place like South Asia, we're also fighting against the environmental elements, right? So, it's a lot of fun. And then the last one I think is very intuitive, but I put this because we get this question a lot of times. Can you get ancient DNA from dinosaurs? And the answer is no, we cannot, because it's extremely old. It's, they lived in the time of 65 million years. The oldest DNA so far, the record holder, is just over 1.2 million years. And that comes from cold and dry Siberia and Arctic region. Okay? So most of the DNA otherwise will be very recent, the last few thousand years, particularly from the tropical regions. Okay? Is this clear more or less? People are getting it? Good. So let's move on quickly. I will skip this part and just go. This is all the repeating. Okay. So very quickly in the next 10 minutes or so to wrap up my talk. Dr. Nagarjuna gave you a lot of different applications of ancient DNA. So I'll just pick a few and I'll give you some concrete examples because we told you like what those applications are. So before getting the human part, I'll just tell you that people have been very creative and used ancient DNA in other kinds of ancient reconstructions. So first would be, he talked a little bit about paleoclimate. take, these are sediment and ice cores. So these can be done in any setting. Obviously, ice cores you can only get in regions where you have permafrozen like Siberia, Arctic and Antarctic. But having done this, one of the studies, I'll tell you very quickly, this was actually a study done by my PhD supervisor doing his PhD work. And what they went to this Greenland. The Greenland is here in the north. They took ice core samples from 4 different places in Greenland. They melted the ice in different parts and they extracted the DNA and they saw what kind of plants and animals they were getting from that. And based on that, I don't have a picture. Yeah, based on that, what they concluded was about 400,000 years ago. Like today, if I tell you Greenland, what do you think? It's all, unlike, it's ice. It's not green. It's, like its name, it's not green. I've been there, I can also tell you. But about 400,000 years ago, it was a very different, it was more sort of like a alpine, kind of like temperate Europe. So they had different kinds of beetles, different kinds of fir, palm trees. So there was the life existing there that was very different from what exists today. So you can actually even look at beyond humans. You can look at what other biodiversity was existing in the past. Particularly if you have extinctions, you can try to understand when mammals don't exist now. But you can actually understand what mammals were like by looking at the ancient humans from these individuals. Yeah, so this is what I wanted to put up. These are mammoths. So there have been studies on mammoths, and that's revealed that actually, when we think about mammoths, we think they are very old, they are extinct a long time ago. But ancient DNA evidence has suggested that they were actually living in North America as recent as about 3,000 years ago in small islands. So they become very fragmented and then eventually they die in those isolated environments. And then the other big, I think, application of ancient DNA has been this hominid. So like I said, we were not alone. Dr. Rai also showed you that tweet. And when you look at small pieces of bones, you may not necessarily be able to say this is modern human, this is Neanderthal. And so this is particularly been an application of ancient DNA because those bones that are not diagnostic morphologically, you can get DNA, compare it, is it within the variation of anatomically normal humans, and if not, then maybe it could be considered something else. So this is sort of how we have developed understanding, particularly of Neanderthals, how they were genetically different from modern humans. And then more recently, some of you maybe have heard of these Denisovans that Dr. Rai mentioned. We don't have a skull, we don't have a fossil for Denisovans. We only have small pieces of bone, particularly this one finger bone. That's how they got the DNA. We still don't know what they look like. But Neanderthals, we have a lot of fossil evidence from across Europe and Siberia. So we have used ancient DNA to identify bones where we are not sure it's an anatomically human or something else. We also use ancient DNA to understand, for example, what is the functional consequences of having Neanderthal ancestry? Is it, what makes us uniquely human? What makes them uniquely Neanderthal? So we can start looking at different biological pathways, depending on the genetic signatures in modern humans, how does it contrast with Neanderthals, and you can start understanding what makes us uniquely human in terms of our susceptibility to disease, our ability to adapt to different environments. Especially this is true because Neanderthals eventually went extinct. So can we, through DNA, make some inference why they went extinct, whereas us, the anatomically modern humans, have obviously become very successful with colonized the entire planet, right? Just giving you a very quick case study of that. This skull belongs to anatomically modern human, so that's us. This skull belongs to a Neanderthal. So if you have a skull, in fact some of the other bones, post-cranial bones, most paleoanthropologists will be able to tell the difference between a Neanderthal and a modern human. And this is Europe. In blue is where Neanderthal fossils have been found. So we know that they existed in all these spaces. So when humans came out of, this is Africa down here, so when humans came out of Africa, we know that these Neanderthals were there right there, right? So it would have been very difficult for ancestors to avoid these Neanderthals. They would have died. So the question then these paleoanthropologists had and they didn't have any way of answering this, is were the Neanderthals who lived there, were they automatically the ancestors of modern people who anatomically were humans? Or did anatomically ones come out and did they wipe out the Neanderthals? Like were we the cause of their extinction? Or did we actually meet with them and we mixed with them? So if that's the case, we should be able to see genetically that we have some mix with Neanderthal DNA. So a lot of scientists, particularly in Germany, have done a lot of this. They've extracted DNA from now many Neanderthals, and they've done a lot of analysis. And what they found is it's the model number 3 that is correct. So our ancestors definitely met, and not only did we meet them, we also biologically interacted with them. And as Dr. Rai said, that all of us outside of Africa, most populations, some African populations also have, but all non-Africans, including us South Asians, we have a few percent of our DNA is from Neanderthals. Okay? So almost 1 to 4% of our DNA is from Neanderthals. So it's interesting to think about that, because I don't know how many of you pause a little bit with this, because first time I heard this result, I thought, wow, so far I was thinking my DNA is human, like anatomically not human DNA. But really no, we have DNA from other sources as well. So I will, is there sort of, I'm looking for time? I'll take maybe a few more minutes and then we'll wrap up. Another cool application has been, of ancient DNA has been natural selection. So Dr. Rai talked about the lactase gene, right, that humans as adults were the only ones who can drink it as milk. Okay, I'll keep talking. But there's another cool example of how ancient DNA showed about adaptations. So you all know in Himalayas it's very high altitude.